Hi everyone, welcome to lab exercise 3.4, which is our last lab of the semester where we will be learning about biotechnology. Make sure that you are keeping all of these handouts because upcoming is our lab final, which is cumulative. It will go over unit 1, 2, and 3, but it's open note and you get to use these lab handouts. Now, today we are going to be identifying different forms of non-disjunction using karyotyping. We're going to label the steps of the PCR process, and we are going to model the use of gel electrophoresis in DNA fingerprinting. First is karyotyping. Karyotyping is a really interesting process because we're going to take a cell sample and artificially induce cell division. So the cell will be artificially induced to start prophase, which means our chromatin is going to condense into distinct chromosomes. But at the same time, we are artificially or chemically going to disrupt the spindle fibers. So what ends up happening is the cell condenses all of its chromosomes, but it never ever separates because these spindle fibers are disrupted. So we can take a picture of this and then organize each of these chromosomes in a predictable pattern so that we can then read and see any kinds of obvious issues of the chromosomes. So the first thing that we're going to do is view the cell by scanning the QR code. So if you scan this QR code, you will be able to see this exercise. This exercise is a cell that is undergoing mitosis, and we have interrupted those spindle fibers and taken a picture. Now what we have to do is we have to organize these chromosomes. So you always have one chromosome from dad and one chromosome from mom. So we're going to find the matching pairs. There are two things that can help us find the matching pairs. One is the size. Notice that the lower numbers, 1, 2, and 3, are very large chromosomes, whereas the ending numbers like 21 and 22 are very small. So what we're going to do is match them based on their size. But for instance, 11 and 12, they look very similar in size. The distinctive difference is the bands. Sometimes they have very dark bands and sometimes they have very light bands. So we will also use the color of the banding as an indicator. For instance, this looks the same size as 22, but the bands do not match. So I'm going to assume maybe this is 20. The same thing here. This looks very large, so it might match 1, but if we hold it up, the banding doesn't match. So I'm going to pull it over here. Whereas this one matches perfectly. The light band, then a dark band, then kind of an alternating pattern at the bottom. So what I'd like you to do is continue to match these chromosomes until you get the completed karyotype. What you can do to help is turn on hints, which can also help you match the chromosomes. Looking at this karyotype, do we assume that this is a male or a female? Well, it looks like we have one X chromosome and one Y chromosome, so I would assume this is going to be a male. Let's look back at our worksheet. And let's view the bottom. It says identify each of the karyotypes as male or female and then identify the non-disjunction order. Non-disjunction happens when a cell is undergoing meiosis, and when it splits, instead of splitting exactly half of the DNA, 23 and 23, because this will end up being a gamete like a sperm or an ovum. Instead of that happening, what happens is we end up with more on one side than the other. So for instance, maybe we'll end up with 22 on one side and 24 on the other side. So if this sperm was selected for fertilization, we would actually have extra DNA. If this sperm was selected for fertilization, it would have less DNA than necessary. So this is a form of non-disjunction, when we get more DNA on one side than the other. And this can cause lots of problems. For the most part, when this happens, we don't even notice because it causes the zygote to die instantaneously. But sometimes non-disjunction can live into adulthood. And let's look at some different examples of that. So the first example here, we have a normal 1 chromosome, normal 2 chromosome. In fact, they all look pretty normal until we get down to the bottom. And so for this individual, our sex chromosomes are XXY. This means that non-disjunction happened in the 23rd chromosome, or the sex chromosomes. Now, the gender of this person is most likely male presenting, but will be somewhat intersex. Okay, this means that the non-disjunction that happened was with the XXY. So this is a form of polyploid. Okay, the syndrome that we're talking about is going to be Klinefelter syndrome. And if you watch the lecture video, we go over these different examples. Okay, so this is Klinefelter syndrome. Some of the symptoms of Klinefelter syndrome will be a reduced size of testes if they are male presenting at birth. But then upon puberty, they may produce secondary female sex characteristics. So it just depends on how male presenting they are at birth. Maybe the family will decide to raise them as a male, or maybe raise them as a female. Now, sometimes the family may not even know, because the genitalia appear very normal, and they may not know that this individual has Klinefelter syndrome until they reach puberty. So most of the symptoms will arise at puberty, where they will get secondary sex characteristics, such as a male presenting will develop breasts upon puberty. Let's look at the second one. The second one looks very normal until we get again to that 23rd chromosome, the sex chromosomes. In this case, the sex chromosomes are XO. We are missing one of the chromosomes from one of the parents. So we're missing one. This is a form of aneuploidy. Okay, that means we are missing some DNA. Whereas the first one we had extra DNA, this one we do not have enough DNA. Now, every single individual with this syndrome will present as female. This is known as Turner syndrome. And some of the symptoms of Turner syndrome include heart disease, a special like skin irritation, skin spots, but the most notable is going to be a lack of puberty. So this female will never have a period. She will be infertile. Her ovaries will not develop fully. Some patients with Turner syndrome may undergo hormonal therapy and develop some secondary sex characteristics, but they will still be infertile. One of the earlier indicators of this syndrome is going to be a webbed neck, meaning that instead of the neck coming down like this, the muscle will flatten, and instead of the muscle coming down the shoulder area, it kind of webs to the arms. So this is one of the more physical early indicators of Turner syndrome. Let's look at the last. The last one looks like we have normal sex chromosomes. So this one has X and Y, meaning it is going to be fully a male, no sex issues, no intersex. This individual is a male. But if we look closely, we have 1, 2, 3 chromosome 21. So This is trisomy 21, which means we have a polyploidy of the 21st chromosome. This is commonly known as Down syndrome. And some of the symptoms of Down syndrome can be a moon-shaped or rounded face, or a higher instance of obesity and heart disease. But generally we think of Down syndrome as a person with a sunny disposition that has a cognitive decline. So they will have a cognitive deficit, but this will generally be in kind of a spectrum. Some persons with Down syndrome need care their entire lives and are nonverbal, whereas others can be very verbal and very independent and even live independently and hold down jobs. So it is a varied, a highly different syndrome where some patients have more severe symptoms than others. But generally, all three of these examples will grow to adulthood. So I wouldn't see any like shorter lifespan with any of these. Let's look at the second page. The second page is talking about polymerase chain reaction. Polymerase chain reaction is a really cool process that utilizes something called Taq polymerase. And Taq polymerase was found in hot springs. So it is a thermal or thermophilic bacteria called Thermus aquaticus. And it really loves heat, or it lives and thrives in heat. And the reason why we can use Taq polymerase for this process is because we heat up our samples really hot and then we reduce the temperature. And that means that every single time we heat up the process, we're going to get double the DNA. Now, this happens in 3 distinct steps. So we're going to go over these 3 steps in a moment. But prior to beginning, we have to have a sample of the DNA along with free nucleotides, a buffer solution, and the Taq polymerase. So when we are looking at this activity, it says label the steps of the PCR using the following image. So 0, prior to beginning, according to this, we're going to use our sample. So we're going to load the DNA sample. The first step is called denaturization. Then it says the second step is called annealing. The third step, if we continue to read, is called elongation. Now, number 2 says write a description of each step. So when I load my DNA sample, I'm going to load the DNA, the buffer. The buffer is going to be a liquid that prevents it from evaporating too much, so we want to have lots of buffer. We also need primers and free nucleotides. The free nucleotides are really important because without the free nucleotides, Taq polymerase would not be able to add the nucleotides in sequence. So we need to have as many nucleotides as we want to make copies of DNA. Number 1 says denature. If we look at this list where it says denature, this is the double-stranded DNA is heated to separate into 2 single strands. So DNA separates. Next is annealing. If we look at the annealing step, it says the temperature is lowered and the DNA primers bind to the sequence. So the primers will bind. And that means that I have, in step 1, separated my DNA sample into 2 separate strands, and in step 2, added a primer to both of those strands. This is going to give Taq polymerase a place to start working, and it'll work in that direction. Step number 3, elongation. If we read our description, elongation is where the temperature is optimized for Taq polymerase, and this synthesizes complementary DNA strands. So this is when we actually copy the DNA. And we're going to end up with double the DNA that we start with every single time we go through this process. And this whole process takes about 1 minute, which means each minute that it's inside of the PCR machine, all it does is raise and lower the temperature. Each minute that it's inside the PCR machine, we're going to double our DNA, leading to an exponential increase in the amount of DNA that we have. Number 3 says note the temperature of each step. So we're going to read in the paragraph and find the temperature that it takes to denature DNA. This is going to be the highest temperature. It takes a lot of heat to break those hydrogen bonds without the assistance of an enzyme. Normally enzyme helicase does this, but we don't have the enzyme helicase, so we have to denature it with heat. This is another reason why Taq polymerase is so important, is because we want an enzyme that can withstand this heat without falling apart. So this is going to happen at about 90 to 95 degrees Celsius, almost boiling, not quite boiling, but almost boiling. Next, the annealing step, we're going to cool it down, but still not down to room temperature and not even to human body temperature, just cool enough for those primers to bind, for those hydrogen bonds to reform, which means this is going to be about 55 to 60 degrees Celsius. And then we raise the temperature again, not quite as hot as step 1, just hot enough for Taq polymerase to start working. So the Taq polymerase works optimally at about 70 to 75 degrees Celsius. Then we will start the process all over again. Every single time we want to make a copy, we will go through this process. If we leave our sample in the PCR machine for about an hour, we're going to copy it or double it 60 times, which means if we have a pretty good DNA sample, we can end up with millions and millions of copies. So let's look at the questions at the bottom. It says, why is Taq polymerase so important? That's because it is a thermostable or temperature stable enzyme, okay? It's a temperature stable enzyme that can copy DNA. So it's essential for this process. What is the purpose of adding free nucleotides? Well, we said that in step 0, or before we started the PCR machine. We need those free nucleotides in order for Taq polymerase to even have anything to grab onto or make a copy with. So Taq polymerase had to have each individual free nucleotide for it to copy. So it needed all of these free nucleotides for it to grab them and pull them onto the DNA. So the purpose is to provide base pairs to Taq. The last question, what problems could arise using this technique if we have a damaged DNA sample? This actually does happen. If we do not have a good DNA sample at this step, then every single time we copy it, we're going to be making a bad copy. If you've ever worked with an old printer and you made a copy of a copy and then made a copy of that copy, it's called the Xerox effect, and you end up with a really bad end result. So basically, copying a bad sample will lead to bad copies. Once we do get our multiplied DNA sample, then we can do a couple different studies with it. So imagine that we did have a good, thorough sample and we put it through the PCR machine. What this does for us is it multiplies our sample so that we have enough to work with, so that we have enough to see. And then we can do a study like gel electrophoresis. If we do a study like gel electrophoresis, then it can tell us a lot about the DNA that we just copied. So step 1 says explore the process of gel electrophoresis by scanning this QR code. I'm not going to scan the QR code, but I'd like you to scan it. What I'm going to do is just look up at the screen. Now, if I read the instructions here, it says number 2, use a concentration of 2% agarose. So I'm going to select my gel and I'm going to select 2%. 2% is pretty standard. It has the most resistance, whereas 0.6% would have the least resistance. So I want 2%, that's what the instructions say. Then number 3 says select DNA sample PBR322. So I'm going to select that one. Number 4, select 1 kilobyte marker. So this means 1,000 base pairs. Then number 5, fill each well with a unique DNA sample. So I'm just going to select a different DNA sample for each well. And then it says to start the simulation and turn the power on. Number 7 says to turn off the power after the lowest marker reaches the bottom of the gel. So now what we have to do is kind of sit here and watch the gel work. So as we are, when we turn on the gel, what that means is that I am actually pumping electricity through the gel. It has a buffer solution that will carry the electricity. And that's why the DNA always moves down, because this is the positive end and this is the negative end. So the electricity is moving from the negative to the positive end. Well, guess what? DNA also has a negative charge. Since it's negatively charged, it will always move towards the positive end or downward. Oh, that one just disappeared, so I'm going to turn the power off. Number 8 says turn on the UV lights and answer the question below. So as I'm turning on the UV lights, it changes what I can see. I cannot actually see the DNA in regular light. What I'm seeing is a dye. The DNA has been modified to have fluorescent markers added to it. So I really can only see the DNA under the fluorescent light or the UV light. Now, the one thing that I do know is well 1 is my marker or my ladder. Normally the ladder will fill up the entire page, but in this example, I only have a ladder for 1,000 base pairs. So let's answer these questions. It says, which direction did the DNA fragment move? Well, it moved downwards or towards the positive. Why does DNA move this direction during an electrical current? It is because DNA is negative, so it always is going to move to the positive pole. The next question, why is the DNA only visible under UV light? It has fluorescent markers, or little tags on the DNA that fluoresce in the UV light. Normally, this DNA fragment is way too small for us to even see it. Next question is, which DNA fragments move the fastest? Well, it's going to be the smaller fragments because this gel has a lot of resistance. Remember, we chose the 2% gel. It has a lot of resistance, and so the larger ones kind of get stuck in the foam of the gel and have a harder time moving around, whereas the smaller fragments move much faster through the gel. So this is smaller. And the next question says, which ones move the slowest? It's the larger fragments. The next question, why do the fragments move at different rates? That is because the larger fragments get stuck. They move slower because they kind of get stuck up at the top. And if I left the gel running, it would continue to pull them down, but at a much slower rate than these. So they actually start all kind of squished up at the top, and as you run it, they spread more and more out. The next question, what is the purpose of the DNA marker or the DNA ladder? Well, this is going to be an artificial stretch of DNA that we always will use every time we run a sample, and it kind of gives us an indication of where each of these base pairs is. Right now I only have a 1000 marker, but if I had a true ladder, it would tell me what each of these locations was supposed to be. So maybe this is 100 base pairs because it's much smaller. So the purpose of the ladder is to indicate the fragment size. All right, next, which sample contained DNA of 1000 base pairs? Well, for that, I'm going to look at the reference. This is 1000 base pairs, and if I look, I have 1000 base pairs here in well 2, which was the ACC one. And then if I continue, I also have 1000 base pairs in well 5, which was eco R something, eco RII. What I'm looking at is this section here. ACC was in well 2 and well 5 was eco RR3 eco RI. All right, the last question, what might happen to the sample if you leave it running too long? They would essentially just fall off the edge. So if we continue to run this, and we can actually do that, we can turn on the power and continue to watch it run, they will eventually all fall off of the edge. And you will have to start your experiment all over again. So it's important to run your sample for an appropriate amount of time, because after everything falls off the edge, then we don't really have any more useful information. So let's turn it off and look at the UV light again. So we've already lost some information down here. All of that fell off the bottom, and you can see more clearly what's going on at the top. So when we look at all of these different answers, which direction did the DNA move? It moved towards the positive. That's because DNA has a negative charge. It's visible because it has fluorescent markers or tags. The smaller ones move faster and the larger ones move slower. That is because the larger fragments get stuck in the gel. The DNA marker indicates the fragment size, and the DNA fragments that are about 1000 base pairs long include lane 2 and lane 5. And if we continue to run it, we're going to fall off. Now, the next section is called DNA fingerprinting. So we are going to use the information that we just learned to do an experiment with our with gel electrophoresis. So in this section called DNA fingerprinting, it says that you are a scientist named Dr. Z and you are recently retired, so you have some extra time on your hand. And you get really annoyed with one of your neighbors because every morning you step out and there's dog poop on your porch. Ugh. So what you do, being a mad scientist, is you collect all the poop. And you follow each one of your neighbors that has a dog and you collect their poop because you want to know who's pooping on your front porch. So let's turn to the last page and see what this means. So this section says, use this chart to complete the gel sample. First, label each lane with a different dog. So let's look at this chart. This chart has the apartment number and the name of the dog that lives at that apartment. Also, it has our unknown poop. So we're going to try to figure out if one of these dogs matches our unknown pooper. It also has a list of DNA fragments, which we will use to run our theoretical gel. Step 1 says to label each lane. So that's what we're going to do first. So lane 1, I'm going to label Fluffy. And that's where I'm going to put Fluffy's information. Lane 2, I'm going to put Bruno. Lane 3, I'm going to put Cupcake. Lane 4, I'm going to put Falafel. And lane 5, I'm going to put Sky. Then in lane 6, I'm going to put my unknown Pooch. And lane 8, you can see, is the ladder or the markers. And in lane 8, we have a marker for 2,000 base pairs, 1,000, 800, 500, 300, and 100. Notice how the smallest fragments move the fastest, so they're at the bottom because the gel is going to pull the DNA down towards the positive end since the DNA has a negative charge. Step 2 says look at the DNA fragments assigned to each dog and using a marker, label the area you think each fragment might move to. We can use the DNA ladder in lane 8 to compare fragment locations. So let's do Fluffy first. Fluffy has a DNA fragment at 760, 390, 1516, 1240, 1190, and 550. So what I'm going to do is using my marker here, I'm going to estimate where the gel will land. So first is 760. So that's almost at 800, but probably a little lower than 800. So I'm going to put a fragment right here. Okay. Then we have 390. So it's a little bit more than 300. So I'm going to put it a little bit above 300. Then we have 1560, which is going to be in between 1,000 and 2,000. So maybe right here. Then we have 1240, which is also going to be in between 1,000 and 2,000, but closer to the 1,000 end. Then we have 1190, which is again like right there. And 550, which is going to be just above 500. So this is what Fluffy is going to look like. Next, we are going to finish and do all of the dogs. So Bruno has 690, which is going to be above 500. Then Bruno has 1340, which is going to be up here. And we have 810, which is right at 800. Then we have 1840, which is almost 2,000. And 430, which is below 500. And 1720, which is almost 2,000. All right, next we have Cupcake, 970, which is just below 1,000. Then we have 1340, which is the same as Bruno. 520, which is also the same as Bruno. They're pretty close. 1780, which is almost 2,000. 670, which is going to be above 500 but below 800, maybe about right here. 1420, which is kind of perfectly in between 1,000 and 2,000. So this is what Cupcake looks like. Let's do Falafel, 1310, 970, 570, which is going to be real close to 500. 1420, about halfway between 2,000 and 1,000. 1840, pretty much 2,000. And 1210, which is going to be a lot lower. So Falafel looks pretty different. Let's do Sky. Sky is 420, which is going to be below 500 and above 300. 1830, which is almost 2,000. 1090, which is almost exactly 1,000. 1430, which is somewhere in between. 720, that's almost 800. And 1670, so right above this line. The last one is going to be our mystery sample. Our mystery sample is 520, 670, 1780, 1420, 1340, and 970. So let's look at our other dogs and kind of compare. And looking at them, I notice that a lot of these dogs have these 4 bands kind of pushed up to the top. But I'm also looking for a dog that has 2 bands kind of at the bottom. So if we look, I think I can eliminate Falafel because Falafel only has 1 band right here. And I can eliminate Sky because Sky's bands are really far apart. I can eliminate Bruno because Bruno only has 3 bands at the top and Fluffy only has 2 bands at the top. So if I were to look for the most similar base pairs, both Cupcake and the Mystery have 4 up at the top in between 2,000 and 1,000, and then 2 down at the bottom in between around 500 to 600. So I would definitely say, who is our culprit? Cupcake. Cupcake is our culprit, and so I'm going to have a problem with my neighbor at 3A. But of course, he probably thinks I'm really weird because I just sampled dog poop all day. I'm retired though, so I have plenty of time on my hand to be playing with these DNA ladders. So this is how some people compare crime scenes. Instead of a mystery poop, it may be DNA left behind at a crime scene, and instead of different dogs, it might be different suspects. This is also how we may do paternity tests, where we will compare the mother and child with potential fathers. And so this is all going to be the same process of DNA fingerprinting. Thank you for joining me this week, and good luck with finals and the end of the semester. Don't forget to complete your group projects.